Good afternoon, everyone. Today, we embark on a journey to demystify the intricacy of past year exam questions for October 2022 for A-level students. Examining the past offers us a compass to navigate the challenges ahead, empowering us with the insight and strategies to conquer the present. Let's delve into the treasure trove of past exam, unlocking the key to success and mastering the art of the academic excellence. Question number 1. The photomicrograph show part of the plant cells. In this diagram, it indicates the part of the plant cells is about 5 micrometer. What is the magnifications of the cells? So the cells in the photomicrograph show part of the cells is 5 micrometer. Therefore, the magnifications of the cells is multiply 4,000. Which row matches each organelle with its function? Its microtubes move vesicle within the cells. Rough endoplasmic reticulum is assembled amino acid to make protein. Golgi bodies, it's actually contain enzyme for destroying wall outside of the cells, and centrolus is moved chromosome apart during anaphase. Where are ATS ribosome found? First, chloroplast. 2, cytoplasm of eukaryotes. Third is mitochondria. And fourth is cytoplasm of prokaryotes. ATS ribosomes are found in the cytoplasm of eukaryote. So the correct answer is option 2, which is cytoplasm of eukaryotes. Which feature, when present in an organism, enable it to be identified as a prokaryote? The feature that enables an organism to be identified as prokaryote is C, which is nucleus, the absence of a nucleus. Number 5, what is the functions of the nucleolus? The function of the nucleolus is the synthesis of RNA, ribosomal of RNA. The answer is D. The diagram shows the result of a number of tests on a solution of biochemicals. We have Benedict test blue, non-reducing sugar test yellow, iodine test, it turns to blue black, and burette test, it turns to blue. Which substances are present in the solution? Based on the result provided, Benedict test turning blue, which is indicate the presence of reducing sugars. Non-reducing sugar test turning yellow suggests the presence of non-reducing sugar. Iodine test turning blue-black, which is indicate the presence of starch. Burette test turning blue indicate the presence of protein. So, based on the test, the substances present in the solution are reducing sugar, non-reducing sugar, starch, protein. Therefore, option B seems to be the correct answer, which is protein, non-reducing sugar, and starch. Number 7, which diagram show the ring form of beta glucose? If you can see that the elements for the beta glucose is a cyclic hemiacetal form of glucose, where at anometric carbon, the position of hydroxylic OH group is in upward direction. If you compare between the 4 diagrams, which is indicate OH group is in upward direction. So A, right? So the answer will be A. Which row correctly matches the example with the type of molecule? The correct row matching the molecule type is D, where disaccharide is sucrose, macromolecule is cellulose, and monomer is fructose, and polymer is a collagen. Number 9, the enzyme alpha amylase hydrolysis amylopectin, but it is not able to hydrolyze some of its glycosidic bond. It's only hydrolyzed 1,4 glycosidic bond, and it is not able to hydrolyze the last bond of a chain, and it is not able to hydrolyze the bond in a chain of 3 units attached by a 1/6 glycosidic bond to another chain. Which glycosidic bond can be hydrolyzed by alpha amylase? The answer given here is C. Number 10, molecule X is a lipid. This is an example of lipid, and this is an example of phospholipid. And which row is correct for molecule X and triglyceride? So, the molecule X is actually contain 1 fatty acid, and for the triglyceride, contain 3 fatty acid. Number 11, bread contains a mixture of polypeptide known as gluten. 2 of the polypeptide found in gluten are glutamine and gliadin. Which statement describes the tertiary structure of a protein? The correct statement describing the tertiary structure of a protein is D. Amino acid with hydrophobic R group are found on the inside of glutamine. Tertiary structure refers to the overall three-dimensional shape of a protein, which is stabilized by various interactions, including hydrophobic interaction, that cause hydrophobic amino acids to be buried inside the protein away from the water. Number 12, a student was provided with 2 test tubes, one containing 10 centimeter cube of solution P and one containing 10 centimeter cube of solution Q. When these solutions are mixed together and left for 24 hours, the concentration of P decreases, but the concentration of Q remains the same. The student wrote the following conclusion. P may break down over time. Q may be a biological catalyst, and P may be the substrate for Q. And which conclusion could be supported by the information? Based on the information provided, the following conclusion could be supported. P may be breakdown over time, and this conclusion is supported because the concentration of P decreases after mixing with Q and left for 24 hours. And this suggests that P underwent some form of degradation or breakdown. P may be the substrate for Q. And this conclusion is supported because the concentration of P decreased while the concentration of Q remained the same. And this indicates that P may have used up by Q, and suggesting that P could be the substrate for Q in a reaction. So the conclusion 2 seems more plausible than the suggestion that Q may be a biological catalyst. And while it is possible that Q is a catalyst, the information provided doesn't directly support this conclusion, as the concentration of Q remains the same and there is no indication of the reaction rate change. The answer will be P may break down over time and Q may be biological catalyst. Which statements are correct for the lock and key hypothesis and the induced fit hypothesis of enzyme action? The substrate is the same shape as the active site. The substrate is held in place in the active sites by temporary bonds. And third, the enzyme and sometimes the substrate change shape slightly as the substrate molecule enters the enzyme. And for the lock and key hypothesis and the induced fit hypothesis of enzyme action, first, the substrate is the same shape as the active site, meaning that this statement is correct for the lock and key hypothesis, where the substrate fit perfectly into the active site, like a key into a lock. However, it is not entirely accurate for the induced fit hypothesis, where the active site can change shape slightly to accommodate the substrate. Number 2, the substrate is held in place in the active site by temporary bond, meaning that this statement is generally correct for both hypotheses. In both cases, the substrate is held in place within the active site by various types of temporary bond, such as hydrogen bond, hydrophobic interaction, and van der Waals forces. And number 3, the enzyme and sometime and substrate change shape slightly as the substrate molecule enters the enzyme, meaning that this statement is also correct for the induced feed hypothesis. In this hypothesis, both the enzyme and the substrate can undergo slight conformational changes to achieve a better feed. However, it is not a characteristic of the log and the key hypothesis, where the active site is rigid and the substrate fit into it without significant changes in the shape. So the correct option would be 1, 2, and 3, which is not listed in this answer. A student investigated the rate of enzyme activity with increasing substrate concentration. The experiment was repeated with the addition of a reversible non-competitive enzyme inhibitor. A graph was plotted to show the result, which graph represents the result of the two experiments. The answer given here is rate of enzyme activity versus substrate concentration, which is applicable to the 4 diagram. But however, if you can see from B diagram, the diagram is a little bit show that the enzyme, the without enzyme inhibitor, the graph is increasing higher as compared to the graph of the line with enzyme inhibitor. The cuboidal epithelium cells has a length of 2 micrometers, and the cell is an approximate cube shape. What is the most accurate estimate of the surface area to volume ratio of this cuboidal epithelium cell? To find the surface area to volume ratio of a cube, we can use this formula. Surface area equals to 6 multiply side length to the power of 2. And volume equals to side length to the power of 3. Given that the side length is actually length of each edge of the cube is 2 micrometers, we can calculate that the surface area is equivalent to 6 multiplied by 2 micrometer to the power of 2, equivalent to 6 multiplied by 4 micrometer to the power of 2, and it gives the answer of 24 micrometer to the power of 2. And in terms of the volume, is equal to 2 micrometer to the power of 3 equals to 8 micrometer to the power of 3. So the surface area to volume ratio is 24 micrometer to the power of 2 divided by 8 micrometer to the power of 3. You get the value of 3 micrometer to the power of -1. And the most accurate estimate of the surface area to volume ratio for a cuboidal epithelial cells, the answer will be 3 to the ratio of 1. This is calculated by dividing the surface area 24 micrometer to the power of 2 by the volume of 8 micrometer to the power of 3, which is resulting in a ratio of 3 micrometer to the power of -1. Number 16, which statement is correct for facilitated diffusion and active transport? The correct statement for facilitated diffusion and active transport is specific molecules are transported across a membrane. Facilitated diffusion involves the passive movement of specific molecules across the membrane through protein channel or carrier, while active transport requires energy, often ATP, to move specific molecule against their concentration gradient. Therefore, both processes involve the transport of specific molecule. Number 17, equal size potato pieces were placed into a test tube and covered with a sucrose solution, and the test tube was left for 30 minutes. All other variables were standardized. After 30 minutes, the potato piece had not changed in size. What can be concluded from this result? The correct conclusion from the result described is the water potential is the same in the potato and in the sucrose solution, and there is no net movement of water into or out of the potato. Since the potato pieces did not change in size after being placed in the sucrose solution for 30 minutes, it suggests that the water potential inside the potatoes is equal to the water potential of the sucrose solution. Therefore, there is no net movement of water into or out of the potatoes, indicating equilibrium in water potential between the potatoes and the solution. Number 18, how many of these processes produce genetically identical cells? Growth of plant's root, replacements of dead human skin cell, repair of damaged muscle tissue, or asexual reproduction of coral. The process that produce genetically identical cells are replacement of dead human skin cell and asexual reproduction of coral. So the correct answer is 2. Which row show the correct number of each component of a single chromosome at the end of the prophase of mitosis? At the end of prophase of mitosis, each chromosome consists of 2 sister chromatids joined together at the centromere. Therefore, the correct row for this answer will be B. Centromere 1, chromatid 2, polynucleotide strength is 4, and telomere is 4. Number 20, which processes occur during interphase? DNA replication, microtubule organization, and synthesis of ribosome. During interphase, the following processes occur: DNA replications and synthesis of ribosome. So the correct processes that occur during interphase are DNA replication and synthesis of ribosome, which is 1 and 3. Embryonic stem cells are able to replicate continuously. What happened to the telomeres during repeated mitotic cell cycles of embryonic stem cell? During repeated mitotic cell cycle of embryonic stem cell, the telomeres generally remain the same length. And this is because embryonic stem cells express telomerase, an enzyme that can replenish and maintain the length of telomeres. So the correct answer is D. They stay the same length. Which nitrogenous bases are pyrimidines? Pyrimidines are nitrogenous bases that have a single ring structure. So the correct answer is thymine and uracil. DNA replication in bacteria was investigated, and bacteria were grown in a medium with only heavy nitrogen, which is indicate N615, until all of the bacterial DNA was heavy. These bacteria were moved from the heavy nitrogen medium and cultured in a medium with only light nitrogen, which is N14. These formed the first generation. The bacteria continued to reproduce, and the sample of bacteria was collected from the second generation and the third, and the DNA analyzed. Hybrid DNA contain heavy and light DNA, which row is correct for the second generation. So the correct options for the second generation would be the option B. The sequence shows a series of bases at the start of a gene. TAC, CGA, CCA, CCA, CAA, CCA, CGA, and so on. After transcription, the mRNA was translated via tRNA into a sequence of amino acids. When this part of the polypeptide was analyzed, it was found to contain the amino acid in the table. What is a sequence of amino acid in this part of the polypeptide? To translate the given mRNA sequence into amino acid, we need to refer to genetic code. The mRNA sequence is TAC, CGA, CCA, CCA, CAA, CCS, CGA. Using the genetic code, we can translate it into amino acid. So TAC goes to NET, which is start the codon. CGA, it moves to ALA, which is 2. CCA is GLY, CCA, GLY, CAA to become ALA, and CCS is actually unknown. I would assume this as another codon for GLY, and CGA is also ALA. So the sequence of amino acid is NET, ALA, GLY, GLY, ALA, GLY, and ALA. So the sequence of amino acid is closer to option B. And however, it seems there might be a typo in a given sequence. If the CCS codon, it means to be another GLY. So the correct answer would be the options for B. The table shows the mode of action of two antibacterial drugs that can affect the synthesis of protein. If bacteria are treated with the drug rimafacin and streptomycin, what will be the immediate effect? One is a transcription will stop, but 40 proteins may continue to be synthesized. 2, if translation has started, protein may be 40. And 3, translation will be inhibited. The immediate effect of treating bacteria with rifampicin and streptomycin would be, first, transcription will stop due to rifampicin binding to RNA polymerase. And 2, if translation has already started, protein may be faulty due to streptomycin causing error in translation. So the correct option would be 1, transcription will stop, but 40 protein may continue to be synthesized. So the answer is only 1, which is not available in any A, B, C, or D. 26. The electron micrograph show a cross-section of a leaf vein and some membrane cell, which structure contribute to mass flow by the use of proton pump. The answer will be C. The diagram shows some tissue types in the plant organ. Which row identify the tissue types? 1 is a folium, 2 is also folium, 3 is also folium, and 4 is a xylem. Which description of xylem vessel element is correct? The correct option is D, which is describe the description of xylem vessels element. There are dead elongated cells with lignified cell wall containing peaks at interval and are associated with companion cells in the root. 29. Which properties of lignin are important for the function of xylem vessels in the stem of a tall plant such as tree? 1, it is a flexible, so does not bind easily. Number 2, it is not permeable to water. Number 3, it is strong to resist collapse under pressure. And number 4, it has weaker adhesion to water molecule than cellulose. So the answer will be 1, 3, and 4. 30. Which row is correct for the sucrose concentration and the water potential in a sauce and a scene in a potato plant at 12 p.m.? So the correct answer will be sucrose concentration, the sauce would be higher. For the scene is also lower. And for the water potential source is lower and the scene is higher. Number 31, the diagram shows one possible way in which sucrose may be loaded into a sieve-filled element. And the answer is a proton move out of the companion cells, which indicate the step which is required by the ATP. And which statement about arteries are correct? The correct statement about the arteries are number 1, number 4, and number 5. Which is number 1, artery walls can resist high pressure. Number 2, the pulse in artery is a result of a surge in blood that causes expansion of the artery wall. And number 4 is the pulse of artery. Okay, number 4. And number 5, there are semilunar valve at the junction of the artery with the heart. So the correct option is C, 1, 4, and 5. What happened during ventricular systole in a mammalian heart? So the answer will be D, which is the ventricular valve closed and the semilunar valve open. 34. A shortage of protein in the diet of children can lead to a disease that causes excess tissue fluid to build up in the abdomen. What explains why a buildup of excess tissue fluid can occur in this disease? So the correct option is a shortage of plasma protein increases blood water potential, less tissue fluid returns to the blood. Number 35, red blood cells may contain a molecule known as 2,3 glycerate, which is 2,3 BPG. When 2,3 BPG bind to hemoglobin, a higher partial pressure of oxygen is needed to bring about 50% saturation of hemoglobin with oxygen. Which statement about the effect of 2,3 BPG are correct? The answer will be 2,3 BPG in red blood cell causes the oxygen association curve to shift to the right, and the binding of 2,3 BPG to hemoglobin lowers the affinity of the hemoglobin for the oxygen. 36. Carbon dioxide is continually produced by respiring cells. It diffuses into red blood cells and is associated into hydrogen ion and hydrogen carbonate ion. Hydrogen carbonate ion then diffuses out of the cell in exchange for chloride ion in the chloride sheath. What is the importance of the chloride sheath? The importance of the chloride sheath lies in the option of B, which is it maintains a balance of positive and negative charge between the cells and the plasma. 57. The photomicrograph shows a section through a bronchus. Which annotated label is correct? Number A is a smooth muscle changes the diameter of the airway. 38. Penicillin is an antibiotic used to treat infection caused by bacteria. Which event explains how bacteria become resistant to penicillin? 1. The event explaining how bacteria become resistant to penicillin are the number of bacteria with the allele for resistant to penicillin increase. Resistant to penicillin is the result of the mutation in a bacterium. And number 3 is bacteria with the allele for resistant to penicillin pass the allele to their daughter cells. So the options for this is B. And 39, which statement explains why some vaccines can be taken by mouth, but tuberculosis vaccine has to be injected. First is macrophages present antigen in vaccine to stimulate an immune response. And number 2, the TB antigen necessary to produce an immune response are protein which would be digested in the stomach and small intestine. Number 14, a student wrote the following statement about the production of monoclonal antibodies. Which statement is not correct? The statement which is not correct is D. T lymphocytes are not fused with cancer cells to form hybridoma cells. Instead, B lymphocyte, not T lymphocyte, are fused with myeloma cells to produce hybridoma cells in the production of monoclonal antibodies. With that being said, thank you very much for your cooperation. And please subscribe to Jom Stabila YouTube channel. And hope that you're doing well in your studies and in your upcoming examination.